Leukemia poses a challenge for cancer researchers. It is a non-solid, blood-borne cancer that is highly influenced by its microenvironment. Current treatments, such as chemotherapy, can eliminate vast amounts of cancer cells. However, leukemia cells have been shown to evade treatments by sequestering themselves in poorly vascularized, low-oxygen, or hypoxic environments in the body. As a result, therapeutic agents cannot reach the leukemic cells, enabling them to survive and grow. One such hypoxic environment where leukemic cells sequester themselves is the bone marrow cavity. Yet, viewing and understanding the process by which cells move into and out of the bone marrow has proved to be difficult. The ability to see into the hypoxic marrow cavity in real time in a living subject would make it possible to study leukemic cell behavior in these locations. These observations would help to direct and advance the development of therapeutic treatments. Much of the current research on acute myeloid leukemia, or AML, has been performed within cell suspension cultures, or ex vivo histological tissue sections at fixed time points. These approaches, while valuable, fail to provide spatial and temporal information about the dynamic tumor microenvironment. In response, the DaCosta Lab at the Princess Margaret Cancer Research Center has developed new techniques and tools that provide a novel way to study AML and other pathological conditions. One platform is the Femoral window chamber, which, in combination with intravital imaging, allows researchers to directly observe cellular activities in the bone marrow over time, within the same animal.
The femoral window chamber is a transparent glass apparatus surgically implanted onto a thin femur of a living mouse. It provides continual optical access into the bone marrow cavity for about 40 days. Using this technique, we can directly observe, record, and track individual cells or populations, such as non-solid cancers, within a living mouse patient. This in turn allows us to track AML growth within the living bone marrow environment in real time. This is achieved by using multiplex fluorescence microscopy that labels leukemic cells, blood vessels, and other tissue components with unique fluorescent color for imaging. When cells leave the circulation and enter a hypoxic environment, they emit a different color of light under the microscope. These engineered hypoxia-reporting cell lines can then be studied within the bone marrow cavity. If these non-solid tumors can indeed evade therapy, then this preclinical imaging platform is the key to determine how AML cells sequester to and interact with the bone marrow microenvironment at the cellular level and over clinically relevant timescales. Let's look at a way that this type of direct observation could inform therapeutic intervention. As an example, we can look at one pathway that seems to be key to the segregation of AML cells into hypoxic environments, the CXCR4-SDF1 axis.
AML cells express many surface receptors, one of which is called CX chemokine receptor 4, or CXCR4. When CXCR4 binds to SDF1, a chemokine abundant in hypoxic bone marrow, it triggers a series of intracellular pathways that activate AML cells' migratory ability. Once activated, AML cells travel along an SDF1 gradient into hypoxic microenvironments that are resistant to conventional treatments.
If we can observe how AML cells display this behavior more closely and in real time, we may be able to develop novel approaches to inhibit the cell's CXCR4 receptors. This could result in AML cells being pushed into the open, where therapy can work more efficiently. The window chamber method will grant us a new way to see and new ways to direct therapy. It will allow us to suggest solutions that interrupt the CXCR4-SDF1 axis and additionally investigate many other pathways that underpin the pathology of AML. Therefore, the window chamber method may improve our understanding of leukemia cell migration within the living bone marrow. It can shine light on non-solid cancer development and unravel how AML cells may use their surrounding microenvironment for tumor growth.
Today, researchers are using the window chamber method to target cancer cells hiding in hypoxic microenvironments by looking at pathways such as the CXCR4 SDF1 axis. However, this platform has the potential to aid in the development of drugs targeting a broad array of diseases. Using the window chamber method, new discoveries can be made to overcome conventional treatment limitations.